Right. So hopefully people have pizza now and we're ready to go. So I'm Nathan Hillson. I'm a scientist in PBD, and I also am a director of synthetic biology at the Joint Bioenergy Institute. And I think what I'm going to do today is definitely talk about this first story, and then depending on how much time we have, I'll get into a second story. So this first story is really about assessing the risks of large-scale metabolic engineering. And this is a really important issue currently as the lab is considering its preferred second campus site at Richmond. So these are the type of issues that are definitely coming up as we interact with the people that actually live in Richmond. So first of all, what do we mean by large-scale metabolic engineering? So for the purposes of the lab, our biggest large-scale facility is the PDU. And I guess Lucas is making a cameo in the picture there, which is nice. So the PDU is located on the floor immediately below J-Bay, so we're all in Emeryville. And the PDU, the largest scale they have in terms of fermentation units, is about 300-liter scale. If you look at the process kind of a flow for the PDU, you start off with biomass. You break that biomass down into sugars, which you then feed to microbes in successively larger fermentation tanks up to about 300-liter scale. At that point, you can separate your biofuel, for example, and then all of the waste is then going to go downstream to a wastewater treatment plant. So when you're dealing with engineered strains on a really large scale, you actually need to abide by BSL 1 large-scale rules. And this essentially says that you have to keep all of your engineered microbes in a contained environment. And you have to actually kill all those microbes in their contained environment before you send them downstream. So at the PDU, you can actually just do autoclaving of the microbial cultures in the very same fermentation units that you're using to grow them up in. There's also some engineering controls. So say, for example, there's no drains in the PDU. They just go straight to the sewage. So any water that basically is spilled in the PDU will go downstairs into the basement where they have these catch basins, which are then base treated and neutralized before sending downstream to the wastewater treatment plant. And all of these processes are validated once a month. So they'll, for example, take the cultures after they've been autoclaved and plate them out and make sure that there aren't any of the microbes that are still alive after these types of processes. So let's just think about a what-if scenario, though. Let's say that for an example, what happens if an engineered microbe actually survives all the way downstream to the wastewater treatment plant? So what are the risks involved in this? So is it possible that our engineered microbe is going to be able to transmit some of its genetic information to the sewage microbial community, or maybe in vice versa? Can the sewage microbes actually send their DNA into our engineered microbes? You can imagine other types of things happening. For example, if you have our engineered microbes introduced into the sewage community, does that actually affect the demographics within the sewage community? So are you going to be affecting the sewage microbial community that way? And not only is it important to think about what are the risks, but what are the probabilities? So it makes a big difference if one of these events happens 1 out of 1 billion times versus 1 out of 100 times. There's also some other interesting questions. For example, can we actually engineer our strain so that some of these probabilities are further minimized? So what can we do along those lines? So I'm going to kind of start out the story by giving you a little bit of an overview of EBMUD. Well, here's kind of J-Bay and PDU up here in Emeryville, and only a couple of miles away is EBMUD. So if you're coming from San Francisco across I-80, then this is kind of the MacArthur Maze. So sometimes as I'm driving to work, depending on the day, you have different odors that are coming off of EBMUD. So I'm reminded constantly that this is where we're getting our microbes from. So if you take a satellite view of EBMUD, you have some characteristic features like the clarification tanks and the anaerobic digesters. We're going to be sampling our microbes from the secondary treatment, so this activated sewage sludge. So just a really quick overview of how wastewater treatment process works. So you bring in the wastewater. Any really big particulates is kind of separated off at the beginning. Then you have kind of mostly like a liquid stream. So then you can get scum like oils and things that you can take off. And then you can do some secondary types of separations. And eventually you're going to have your liquid flows going into the secondary treatment, which is microbes that are growing in an aerobic environment that's actually going to be processing the wastewater. So this is where we're actually going to be sampling our microbes from. So you can actually take a field trip to EBMUD. So basically, you can take a little tour there, and it's actually a really worthwhile and interesting thing to do. So this is our tour guide for the day. This was Carol, and this is just to show you that sampling wastewater or the sewage sludge is really not a highly technical endeavor. So I'll just show you just a quick movie of Carol just doing just a really quick sampling of some of the sewage sludge. So she has basically just an Nalgene bottle at the end of a PVC pipe, and she'll just stick it into the wastewater there and bring it back up. So that's pretty much as sophisticated as our sampling gets. And the sludge samples, so if you swirl them, you have all these suspensions of the microbial communities, but they actually flocculate and settle down pretty quickly. And if you look at these microbes under a microscope, you can see they're highly diverse communities. So it's not just one single type of microbe. It's a whole mixture and community of microbes. So we actually want to know on more of a molecular level what's actually contained within the sewage communities, and you can use phylochip analysis to do this. So you can get a distribution of the different types of microbes that you would find in this sample. So this was from July of last year, and oftentimes you see these populations dominated by proteobacteria. So now we want to know, can we actually culture these communities in the lab? So we have a mock sewage reactor set up. And essentially what we have is 3-liter reactors that are aerated and continuously stirred to kind of mimic what you'd be finding in the secondary sewage processing at EBMUD. And we have a defined media, which also tries to mimic the wastewater. So how good of a job are we at actually doing the culturing? Well, this was kind of our initial microbial community from July. In our first culture, and we preserve a lot of the diversity, and then in the subculture, we can also preserve a lot of that diversity. So it looks like we're doing a pretty good job. And just in comparison, if we actually sample the sewage community on a different day, so this is about 3 months later in October, there's actually more variation from the July to the October sampling than we saw variation when we were actually growing these cultures up in the lab. And if you talk to Carol at EBMUD, she'll actually tell you there's almost day-to-day variation. So if you sample in the middle of the week, it's going to be very different than on a weekend. Or if you sample right after the Super Bowl, you're going to have an extremely different community than you would maybe during a normal work week. So then how do we kind of set it up so that we have our sewage communities exposed to our engineered microbes? Well, one of the ways that we can do this is through a hollow fiber module exchanger. So essentially we can have our sewage community spatially separated from our engineered microbes. And essentially what you have is you have the engineered microbes kind of flowing along this kind of red arrows, and they're kind of flowing outside of these cylinders. And then inside of the cylinders, in a countercurrent fashion, are the sewage community. And although the sewage community and our engineered microbes can't actually physically touch, they can exchange DNA and other types of solutes. So what we're really trying to measure in this type of an experiment is, can we actually measure any DNA getting exchanged between our engineered microbes and the sewage community? So generally when you try to do these experiments, you try to give the sewage community an incentive to actually pick up the DNA from your engineered microbe. So normally in the lab, what you might be thinking about doing is using an antibiotic resistance marker. So you would give the sewage community the benefits of being resistant to an antibiotic if they take up this DNA from our engineered microbe. The challenge is in the sewage, they're actually resistant to every antibiotic that you can throw at it, which kind of makes sense because people are at home and they're taking antibiotics, and then they go to the washroom and that all ends up in the sewage. So we can't use antibiotic resistance markers. So we have to come up with other little carrots that we can dangle to try to get the sewage community to take up our DNA. So one of the ways that we've pursued trying to identify these carrots is through a tool called GLAMM that was developed by John Bates and Adam Arkin's lab. And GLAMM kind of looks like Google Maps, except instead of like trying to get from San Francisco to Berkeley, you basically try to get around cellular metabolism. So this is basically all of cellular metabolism, and not every single microbe can actually go through all of these routes. So if you look at E. coli, for example, instead of all the routes that are being possible, there's only a subset of pathways and nodes that are actually available to E. coli. So what we wanted to do in terms of a carrot is identify nodes that are just one step away from central metabolism that normally E. coli cannot process. So the idea would be we're going to feed E. coli a food that it can't normally eat. And then if we introduce one additional gene or one additional enzyme, now it can eat that food. So we were going through GLAMM, and we identified one particular node, and it turned out to be chloroacetate. And it turns out that if you just introduce one single enzyme, one single enzyme, you can actually get from chloroacetate into central metabolism. So now we want to see, can the sewage microbial community already eat chloroacetate? So what we did was we grew the sewage community either in the presence of just glucose, which is just a normal sugar food source, or in the presence of different concentrations of chloroacetate. And what you can see is there's a delay in growth between the sewage community growing on the chloroacetate and on glucose. And that corresponds with the change in the microbial community. So if you're growing the community on glucose, you continue to see a very diverse set of microbes. But if you grow it on chloroacetate, it turns out, or our hypothesis is that only a small subset of that community can actually grow on chloroacetate. So basically, you start with a really diverse culture of microbes, and only some of them can eat chloroacetate, and that's kind of the population that you end up with once that population has a chance to basically entirely take over. Another interesting point here, though, is that just introducing more and more chloroacetate doesn't result in more and more growth. So if you start feeding increasing amounts of chloroacetate, eventually the community is going to get to the point where more chloroacetate doesn't result in more growth. So not only can chloroacetate kind of be a food source, but it also can have almost like an antibiotic effect, where basically if you feed in too much, then it actually is growth inhibitory to the microbes. So this is going to be a really nice carrot for us. So now the question is, which genes do we actually pick to get us from chloroacetate to the central metabolism? So there have been several previously published studies that identified a couple of different genes from different organisms that can actually be expressed and are functional in E. coli. So what we did was we took 4 of these different types of candidates, and we got them synthesized either in kind of their normal codon usage or codon optimized for E. coli. And this is new data. This is just in the last week. But what we've done is we've put all of these different genes into a variety of different expression systems in E. coli and tested them out. And it actually looks like some of the gene candidates that we have are actually pretty effective in converting chloroacetate into glycolic acid. So our next steps are basically to see if we can actually get our E. coli to actually just grow on chloroacetate alone as its food source. We've been also pursuing other methods for identifying carrots. So this is an OmniLog. So the idea is that in parallel you can grow your microbes on many different food sources. So all the food sources are listed here. And if you see kind of a growth in the curve going up, that means that they're able to metabolize it. And if it just kind of stays flat, then they're not able to metabolize that particular food source. So you can compare the sewage community versus E. coli. And what you really want to find are food sources that neither E. coli or the sewage community can actually grow on. So this is a way of identifying that maybe D-threonine is going to be a good choice for us to use. And then a third carrot that I'm not really going to be talking as much about today is actually kind of came out of a collaboration through JBay. So when we're actually deconstructing biomass from the actual biomass itself into the sugars, one of the processes for doing a pretreatment is using ionic liquids. And it turns out that ionic liquids are actually very growth inhibitory to microbes, almost like an antibiotic would be. So it also turns out that the ionic liquids is also extremely growth inhibitory to the microbial community. But it's been identified at JBay pumps that can actually pump out the ionic liquids from inside of the cells. So it's almost like an antibiotic resistance mechanism. So we're also thinking about an ionic liquid pump as being another type of a carrot that we could use. So our next steps for this project are really to start quantifying the horizontal gene transfer from our engineered microbes into the sewage community. And following that up and figuring out which of the microbes in that community are the ones that are actually picking up our DNA. And we can also start to think about which modifications can we make to our strains to start minimizing the risks of these different outcomes. And we're also thinking about collaborating with other researchers that are developing other types of risk mitigation strategies, such as kill switches, to try to make the environmental containment of these strains much more strong. So I think with that, I'm just going to acknowledge people from this first project. So Jill Geller and Sangwan and Dan are really the leaders of this project doing the work. We've had a lot of other assistants from Monica and Cindy, Mary, and Tomas. And I'm a co-PI along with Terry and Adam. I'd like to thank Harvey for some of the slides from the PDU. Carol gave us the tour of EBMUD. Lauren and Francine were helping us with some of the phylochip analysis. John gave us some suggestions about GLAMM. Edward helped out with some of the HPLC analysis, and our funding comes from the DOE, and our project manager is Libby White. And I think what I'm going to do now is I'll be happy to take any questions from this first part of the talk, and then after maybe we do take a couple of questions, that'll move on to the second topic. So if anybody has any questions they'd like to ask now, feel free, or if you want to hold off until the very end, you're welcome to do that as well. Thanks. Right. So Heinz's question is getting back to the very beginning of the talk, and one of the questions that I posed are what are the actual probabilities of these transfer events happening? So what we're going to be doing once we actually start doing some of the qPCR experiments, or quantitative PCR, and also some of the FACS methodologies, we can actually start to assess how many microbes are actually getting the DNA. So we can start to get to some of these transfer numbers. Now, if you actually want to start comparing the probabilities in our mock reactors versus what was actually happening in EBMUD, then we might not have numbers that exactly correspond between the two. But you can start figuring out how many events there were in transferring the DNA from our engineered microbes into the microbial community. So these methods for detecting horizontal gene transfer can be quantitative. So you can start getting quantitative numbers on how often these things happen. So the question is if there's actually precedence of microbes transferring DNA, especially in the context of the sewage microbial community. So there have been lots of studies over the last decades investigating horizontal gene transfer and their mechanisms. I'm not as familiar with the specific examples of that happening inside of the sewage microbial communities, but that would be a good baseline to check against. So maybe the rates of transfer between E. coli and the microbial communities is a little bit less because maybe it doesn't have all of the same conjugative transfer mechanisms and those types of things. But that would be the important baseline to compare against.
How long have you been able to keep your reactor sewage community going before it crashes or does something weird? And have you tried mixing in your engineered E. coli directly into that community to see what happens?
So there's two questions. The first question is how long are we able to propagate the sewage communities inside of our reactors? So generally I would say most of our runs, their duration is less than about a week. So that's probably about the time scale that we've done. We have done serial cultures. So for example, you can inoculate once, grow for a week, and then take an inoculum from that and then regrow it. So in the kind of the middle of the talk, I showed kind of how the diversity changes from like the initial growth to basically a subculture. So we've done that at least a couple of times. What we really prefer to do is go straight from the sample from EBMUD, so we actually have the best representative microbial community, because as soon as you expose the sewage community to glucose, or our mock media for the sewage, then you start to see a slight change in the demographics. So most of our experiments we want to do basically as quickly as possible from our original stock. So they actually have big, almost like balloon bags of the culture that we use as our inoculum. So that's generally how we go. But you can propagate them for on the order of a week and then re-inoculate. And then the second question, you'll have to remind me real quick.
Have you tried putting your engineered microbes?
Oh yeah. So that's right. So the question is, have we tried to expose our microbes directly to the sewage community? And so I showed earlier that we have this hollow fiber module exchange where you keep them physically separate. But we are going to be putting the microbes in directly with the sewage microbial community, and we're also thinking about just taking our purified DNA and putting that straight into the to the microbial community. So we are going to be doing those types of experiments as well. Yeah. Okay. If there's no other questions now, I think we have time to start up the second part of the talk. So this is more the research that goes on with JBay funding. So this is going to be around working on enabling technologies specifically towards DNA assembly, so both the software and the devices. So I'm going to organize the second part of the talk kind of around the synthetic biology design, build, and test cycle. So let's say that we want a microbe that's going to be making a biofuel for us. So we have a set of different, perhaps, plasmids and enzymes and coding sequences and promoters and terminators, and we have to select those components that we're going to be putting into our microbe in order for it to be able to produce our biofuel. So you have a repository of your components. You're going to be selecting some of those components. You're going to be putting them together into a DNA construct. And then once you have that inside of your microbe, you can actually test, did I make the biofuel that I wanted to make? So that's pretty much the design cycle. So you pick your parts, you build it, you test it, and then you learn from that testing. So basically, after you do the testing, you can figure out, I might want to tweak it in this other slightly different way to try to even make more. So this is kind of the cycle that we'll be talking about. And the first part I'd like to touch on is this component repository. So when you have an institution like JBay that has hundreds of researchers, it's really not sustainable to try to store all of your DNA sequences and all of your plasmids and strains just in Excel sheets. There's problems that you have postdocs that are only at JBay for a few years, and after they go, who knows what happens with these Excel sheets that might just disappear on their laptops. So we really wanted a much more sophisticated, institutional level piece of software that could actually maintain all of the information about our strains and our plasmids and our sequences. A further reason why this is extremely important is that the Bay Area is susceptible to earthquakes, and it could be the case that if we have a really big earthquake, then maybe our power is going to be shut down for even a week. And if that's the case, then even the emergency power for our minus 80 freezers is probably going to go out, and it's possible that we could lose all of the strains that we have at JBay. So one possibility is that you could actually rent a facility, for example, in Iowa, so somewhere far away from California, where you have almost like a replica copy of all of your strains. So we looked into that possibility, but it turned out that it's much more expensive to actually store all of your strains in minus 80 freezers in Iowa than it is to just maintain all of the DNA sequence information. And then if you lost everything, then just to get a DNA synthesis company just to remake the sequences that you actually want. So as a strategic decision at JBay, we've decided we're not going to be housing a replicate copy of all of our strains offsite. But basically what that means is now we have a tremendous amount of pressure on our informatics division, because if we lose the data about all the sequences and we have a major earthquake, then we basically have nothing. So there's an extremely important reason why we have to have really good data. And then here, not only do we have data, say, stored at JBay, but it's very easy to have offsite data storage. So that's how we're tackling this. Now, when we first started, there really wasn't any commercially available software or even academically available software that was going to be able to do this for us. Probably the best system in place is the MIT Registry of Parts. So it's actually very well set up to store lots and lots of sequences and strains from people all around the world. But the challenge with the MIT Registry was that as soon as you introduce a sequence or a strain into the MIT Registry, everything is public. So there's no way of keeping your sequence information private until you publish, so you could very easily get scooped. Or for intellectual property reasons, it's also not a very good idea to put things into the MIT Registry. So then we began to develop our own repository, and I'm just going to show you a brief demo of it because it's easier to show than just to show in screenshots. But just to know, it was recently published, and it's web accessible. Anybody can sign up with just an email account. It's also completely open source software, so it's under a BSD license, so anybody can basically further adapt it for their own needs. So I'm just going to break out and start up a quick demo video of it here. So let's get this guy going. And, all right. So this is the ICE repository platform, and this is at JBay. And this is a new version of the software, so it was just released a little bit over a month ago. And all of the data inside of the ICE repository is organized into collections. So at JBay, we have about 25,000 different entries currently in the registry. And some of these are in just kind of really big collections, for example, an E. coli deletion collection. But each user actually has their own sets of collections. So here's one of my collections, which is basically just all of the sequences and strains associated with one of my papers. So we can create new collections, so maybe we'll just create a new collection. And right now there's no entries in that collection, so maybe we just want to add a particular strain to that collection so it's not so lonely. And now we're going to have just one strain in this new collection that we created. And if we want to, we can also remove strains and sequences from those collections, and we can completely get rid of this collection if we'd like to. So it's pretty easy to create your own personal collection, put sequences or strains into it, take them out. If we just go into this collection from my paper, we can sort all of the contents of that collection by type. So maybe you want to do parts and plasmids first. And the information contained about each one of these entries is things like what is the name of this entry, a summary of what it is it complete or is it in progress, does it have sequences attached, and so on. We can just hover over any of these links, and it'll give us a quick snapshot of what's actually contained within that entry. And we can click on any of those entries and get more information about what's contained within it. So in these entries, we might have information, for example, about who the principal investigator is, what the funding source is, the biosafety level for plasmids, all the strains that it's contained within, additional information, for example, summaries, information, which papers are associated with this particular entry, et cetera. So the other thing that we can do is actually associate a DNA sequence with this entry, and we can download it if we'd like to. So we can download it into kind of standard formats like GenBank or FASTA, and the new synthetic biology open language format as well. At the very bottom, we'll actually just see a sequence map of the DNA contained within that particular entry. And if we'd like to see this more on a DNA base pair level, we can just open up this sequence in Vector Editor, which is also freely open source software that we developed. And we use the software mostly for editing sequences or annotating the sequences. And once you make changes here, you can just save it directly back to the registry, which is nice. There's some other functionality that's built into the software. So, for example, if you want to see where DNA restriction cut sites are located, you can do that. There's also kind of a cute little tool that you can play around with, which is essentially a virtual DNA digest simulator. So we can basically say, what would a DNA digest gel look like if we digested this piece of DNA with NDE1? So you can see kind of like a virtual simulation of what that banding pattern would look like. So maybe now what we can do is just maybe click this particular GFP variant. So it has a couple of mutations within it, and maybe we'd like to know which other entries in our registry have these same mutations. So we can just click this piece of DNA, so this feature, and we'll copy it, and then we can go back to the registry. And then we can scroll back up to the top of the display, and we can do a blast search. So we can basically search every entry in the registry for sequence matches to that particular sequence. So we can do a blast search where we just paste in our sequence. And we can also, at the same time, search for other types of criteria. So say, for example, we want to find this particular sequence, plus we want it to be in a plasmid that has chloramphenicol resistance. So we can also say that this particular construct should also contain a chloramphenicol marker. So we'll just add that filter and do our search. And then it'll bring up the resulting constructs. So a couple of other features to show. So once we get the results from the blast search, we can see which sequences were the perfect matches and which ones match a little bit less well. But if we go back into this entry that we were looking at before, I also just want to show a couple of additional features here. So there's a sequence analysis tool. So when you're constructing DNA, you often want it to actually validate that it's the DNA that you're trying to construct. So what you can also do in this tool is upload any of your sequencing traces from Sanger sequencing. So these are ABI files, and it'll overlay the sequencing trace against your plasmid map. And where it's green, it's good, and where it's red, there was a mismatch. So it's pretty quick to visually assess, is this the actual sequence that I was trying to make? And it's also really nice because you store all this information with the actual entry in the registry, so you don't lose any of that information. We can also attach files, and we can control permission. So right now for this sequence, anybody at JBake can read it, but only I can modify it. So you can add people that can read it and modify it if you'd like to. Now we talked about entries, but just quickly show how you can actually create them. So you can create them either through kind of standard web form entries, or you can import a whole bunch of sequences at once. So maybe if you're a postdoc and you're about to leave JBay and you have to put in a whole bunch of plasmids at the same time, then you can do hundreds at once. And this is pretty much just like a spreadsheet. So you can create your list in Excel, paste it in here, just upload your sequences and the attachments and zip files, and then in one go you can basically create all of the entries that you needed to create. And just one final level of functionality is that in addition to just all the informatics, we also store the physical locations of these in the registry as well. So if we go to our E. coli deletion collection, the KO collection, we can click on one of these strains and then click on the physical samples, and it'll show us where exactly in JBay are these strains located. So we can see we have a working copy and 2 backup copies. And for the working copy, it shows like which tube number it's in, which well of a plate, and which type of a collection it's in. So that's just going to be the end of the demo. So now I'm going to go back to PowerPoint. And this is basically where we were in the demo. And just to point out, we're actually using these 2D barcode tubes at JBay. So not just trusting somebody to write with a Sharpie marker. Sometimes it's completely intelligible. And if you go back 2 years later, like who knows like what it actually says. So we're storing all of our strains now in these 2D barcoded tubes. And in order to use them, you really do need to have these scanners. So we have 2 different scanners set up. So one scanner is set up to just read a single tube at a time. Or we also have a plate scanner where you can read in a whole plate at once. And the nice thing is once you scan in a tube or a plate, it's actually linked with the registry. So you can actually know exactly what's in that particular tube. And oftentimes in our plates, we actually get the strains put back in the wrong slot. So if you actually put your sample in the wrong place, then it automatically updates the registry so that the locations are updated as you make the scans. So the other really kind of cool thing about this repository, since it is open source, and since it's a really useful tool, other places around the country and even the world are starting to use it at their own institutions. So this is kind of the JBay registry. We also have a public version. So generally what we do is we keep all of our strains private at JBay until they're published, and then after they're published, we push them just to the public registry so then anybody can see them. It's also been picked up by institutes in Korea. It's the registry for SynBerk, which is the Synthetic Biology Engineering Research Center in the U.S., funded mostly by NSF. And it's also been picked up by the University of Cambridge. And these are just the institutions that we actually know about. We don't really keep track of who's actually downloading the software and installing it, so there could be other instances as well. So that's pretty much the description of our component repository platform. So now I'll just talk briefly about some of the design and build portions of our tools. So if you're a biologist and you go into the lab, you often just will go up to a whiteboard to kind of rapidly sketch out what you actually want to be making. So you might be sketching out several different GFP variants with a couple of different localization tags and linkers and degradation tags. And we really wanted to replicate this experience on the computer. So we developed a computer-aided design tool, device editor, that really just replicates that experience. And this has been recently published. And then after you actually know which parts you want to be putting together, now you actually have to figure out how am I actually going to go into the lab and actually construct these things together. So this is another tool that we've developed, J5, which designs the protocols for you automatically. And it uses 2 different styles of methodologies. And the important thing in J5 is it's one of the unique tools that can actually put together all of these pieces of DNA in one pot and give you exactly, at a base pair level, exactly the DNA sequence that you'd like. And it's also one of the only tools that can do this in a combinatorial type of a fashion. So oftentimes in biology, we don't know what is the best design to use. So oftentimes we want to build many, many different variants of a particular design. So maybe we have many different variants of a particular enzyme or a different promoter and so on. The other thing that J5 does, which is really nice, is it actually does the cost optimal design. So it basically compares how expensive it is for you to make a portion of this construct in-house versus just getting it outsourced to a DNA synthesis company like GenScript or DNA 2.0. So it'll tell you exactly the cheapest way for you to go about and making these things. And that's another unique feature. So just for the purposes of time, I'm going to skip the demo of Device Editor and J5. But if you'd like to, there is a link to the same video demo that you can just see. So if you're interested in seeing the demo of the software, you can just go to j5.jbay.org. So after you've designed all these protocols with J5, the next thing you want to do is go into the lab and do the PCR for these. So if you're putting together many DNA constructs, potentially you have many different PCR reactions. And we're going to be doing these in 96-well plates. And at JBay and many other institutions, we have these thermocyclers for which you can set the annealing temperatures to different zones. So maybe we can go from a cold annealing temperature all the way to a hot annealing temperature. And we really want to match the temperature of the annealing of each PCR reaction with where it should be in the plate. So J5 will also automate the design of where you should be arranging all of these PCR reactions. Now, you could go ahead and do this all by hand, but if we have all this information in the computer already, why not just have a robot do it? So that's kind of the next step. So one of the big challenges we have at JBay, and it's not specific to JBay, it's very common to any research institution, is that we have some very nice robots, but they're almost never used. And the question is, why are they never used? And part of the reason for that is that oftentimes it takes longer for somebody to program the robot to do what they want it to do then it actually saves them time in pipetting. So until that equation is favorable in terms of the researcher, people just aren't going to use the robots. So Gregory, who's here in the audience, and Nina have been developing a robotic programming language that's at a very high level and biology friendly. So essentially what this looks like, this is for basically making breakfast drinks. So we would define basically, where is our drinks plate located, and we're going to define our components like our water and our tea extract. We're going to say how big our water volume is going to be, and we give the instructions of how to make these different recipes. So maybe for chai, we use tea extract and a bit of water. For coffee, we use bean extract and a bit of water. And then we can just tell the robot to make us these drinks. So that's the type of level of sophistication that you have to have just to basically get the robot to do what you want it to do. You don't have to tell the robot, move this pipette from XY 100 0 to the other location. It's really not that complicated. So in a very short amount of time, you can get the robot to do what you want it to do. And this is kind of the web interface for Parpar. So essentially, you can just type in your script here. You can select from a variety of different robotic table layouts. And if you want to, you can actually just preview what that table layout looks like. So in this particular example, we have a series of 96-well plates, some 24-well plates, and some troughs. And this is pretty much the entire interface. So you just press Prepare Robot File, and then you can run it right away on our TCAN robot. So that's actually making work at JBay a lot easier. So robots are great, but they have problems that they have minimum dispensing volume. So every time you dispense with a robot, the smallest size is about 5 microliters. And I think the real future, especially for DNA assembly, is going into microfluidics. So the idea is that we can develop these microfluidic devices that use much less reagent per DNA assembly reaction. So just to kind of give you an example of how this would be laid out, so maybe we're going to have 8 different pieces of DNA and a vector backbone that we're going to be putting together. So we can have little droplets generating for each of these different sources. Then we can focus these droplets, so if they're kind of coming in one at a time, we can focus them in this droplet merging chamber so that now they're just one big droplet. And sometimes it works really well, and sometimes you might have improper mixing. So I'll just show a quick video of kind of what this looks like. And this is in collaboration with a NUPS group at JBay, and he's from Sandia National Labs. So this would be kind of the droplets coming in, and they can merge in this particular channel. We have some other ways of merging droplets also that I won't be showing, but that's one of the methods that we're thinking about. And I'll just arrest that. And then just the next thing to show is what happens after you merge the droplets. So after you have this chamber where you're mixing the droplets, then you can put them through these little undulating S-curve types of mixing routes, and then you can have them kind of do a sinusoidal kind of a holding pattern, so that if you have to do any incubation steps, that's pretty much how you kind of keep them going through the chip. So we have some blueprints for some designs that can do thermal cycling between different temperatures. We're even thinking about feeding in competent cells and doing the transformations on chip. So all of this technology is probably something like 4 to 5 years away. But even today, we're starting to do our preliminary tests of actually seeing if we can actually get DNA assembly to work on a microfluidic scale. So tying this all together, how would this all look like? So you might be putting together a pathway design for your biofuels. You could use Device Editor to arrange all of the components. J5 will tell you how to actually go in the lab and construct them. We could use Parpar to drive our robots to put together all of our PCR reactions. We could then feed all of those DNA pieces into a microfluidics chip, and then out would come all of our cells, and then we can go straight to mass spec types of assays or microscopy. So pretty much in the second part of the talk, I've told you a little bit about our repository platform, some of our design tools, our robotics and microfluidics. And I haven't really spent much time on the actual analytics, but this is a really important part of the process as well. And I think with that, I'll just go to some acknowledgments for the people that developed all of the software. So JBay ICE was really pioneered by Zenovi and Tim, and Hector has taken over. Device Editor is mostly developed by Joanna. Rafe was a postdoc in my group that helped with a lot of the experimental validation of J5. Gregory and Nina have developed Parpar. A NUPS group, including Chiep Chang, who's now at Slack, and Jess have been doing the microfluidics. Jay for a lot of just guidance and support. Our funding has come from Department of Energy and a Berkeley lab, LDRD. And a couple of these acknowledgments are due to the demo that I didn't show you, so I won't dwell on these. And I'd also really like to emphasize Robin Johnson's help in tech transfer. She's been really wonderful. And the J5 and device editor technology has actually gotten licensed and is the basis of a startup now that has 8 employees at the UCSF Mission Bay campus. So that's been also kind of a really exciting thing for me, that founding a company and starting to see the commercialization of some of the work that we do at the lab. And with that, I'll just kind of show a slide of where people are registered for J5 around the world. And with that, I'll be happy to take any questions that you all have. All right. Well, if there's no questions, that means I can get some pizza too. So, all right. Thanks.